you can see, I've recently moved to a new setup, the A-Star Infectious Diseases Labs, and this was an outcome of the transformation or reorganization of A-Star in the past 2 years. So where they have sort of evolved new programs and also right-side various research groups to look at different research questions. So over at the Infectious Diseases Labs, we have 3 main pillars, which is on vector-borne diseases, antimicrobial resistance, and respiratory diseases. Now, the key interest is to build on concepts and to understand mechanisms, pathogenesis of various diseases, and how could we then pivot to some of the preparedness platforms when the need calls for. Now, for my own research group, I think the chairperson has kindly introduced what I have been working on since 2007. And what you will hear today is the work that my team and I have been working on chikungunya over the last 15 years or so, around 12 years or so. So a lot of it is going to be based on animal models to understand the various inflammatory pathways and how could we target some of the immune cells and also the specific pathway to control infection and disease. Just a little bit of an introduction. Well, since the re-emergence of chikungunya in 2006, you could see that it has actually spread throughout the whole parts of the world. It started off from East Africa, which then spread it to Southeast Asia, and then later on in 2013, late 2013, it then went to the South America. So coming back to the animal model, as I was mentioning, because the earlier part of the work was actually spent building clinical collaborations with the team at NCID to understand the disease. So along the way, my team also established the relevant mouse models in order to further understand the deep mechanisms of this virus-inducing inflammatory disease. So this was a mouse model that the team readapted from the wild-type C57 black 6 animals that was first reported in a group in Australia. So you could see from here that all the animals will recapitulate the joint swelling effect, as you could see from here, and also had high levels of detectable viremia. So I'll just put this in summary, using this model that really enable us to answer various questions from innate immunity to the specific immune subsets. And up to this day, we are still asking questions that we are based on some of the earlier findings. So I will now move on to some of the latest, the more recent studies that the group have been working on, and some of these are in collaboration. So one of this is with the collaboration with Andreas Merits from Estonia, where using another alpha virus, the Semliki-Forest virus, his team actually first found that potentially by creating some mutations in the non-structural protein of the virus actually disrupted the viral kinetics. And so the discussion was then, what would the effect be like in chikungunya, since many of these alpha viruses are fairly similar in terms of their molecular organization and also the disease phenotype. So you could see specifically there were 2 key mutations, one in the between the NSP1 and NSP2 region. So then what my lab did, which was done by a former PhD student and now postdoc who has since left and is currently in Rockefeller University now, was to define the mechanisms of these mutant CHIKV viruses. So you could see from here with the different mutation, so the black line represents the wild type without any mutation, and you could see from here that specifically the one in red, the RH mutant, and also the RHEV, the double mutant, actually showed very high reduction in the joint swelling, as we have measured in animals, and also the reduced viremia during the early days. So what he then did was to actually have a deep analysis of the immune cell types in the infected joint. And with the high dimensional analysis of this in the joints. So specifically, he found that one population of cells, which is the inflammatory monocytes, represented by this cell type CD11B+, CD64 positive, and the Ly6C high, that were specifically different. So just to make a long story short, I think the interest was if these mutations, mutant viruses could also act as a vaccines. So you could see from here, this is the regime where we first inoculated the animals with the mutant viruses, and 3 months after, we challenged them with the wild type virus. And all the animals actually did not have any detectable levels of viremia, and neither did they suffer from any joint swelling. And along that time when this work was being completed, then there was another interest of a very close relative of chikungunya, which is called O-nyong-nyong virus, ONV, which was also first documented in Africa, specifically in Uganda back in the late 50s. Like chikungunya, this virus will also cause joint inflammation. And so then there was a resurgence, new cases back in 2017. So it was around that time that we should expand this particular universal chick vaccine. So we wanted to see if these attenuated viruses would also cross-protect against ONV infected animals. And the outcome was that yes, indeed, all the vaccinated animals did not have any viremia in their blood, and also had low levels. I mean, they did not suffer from any joint swelling at all. Now then I'll move on to some recent work. I would like to introduce some of the various members. So while looking at the pathogenesis of CHIKV, there's also the interest to look at host-pathogen interaction. So one of these earlier work, which was done by Guillaume, who was in my lab previously, and he uncovered a particular host factor called VCP P97 as a potential pro-viral host factor that could actually enhance the replication of the virus. Now, so since he has just recently spin off into his own lab to continue this line of investigation of host-pathogen interaction, how could some of these host factors be a target to control the virus, both in vitro and also in vivo. So one of this was, while continuing the question with interaction of VCP, he assessed that if we were to look at to control the post-translational modification of this host factor, specifically on the trimethylation, by targeting the trimethylase, could he actually control the virus replication in this case? And you could see from here that indeed, as an in vitro data in cells, he was able to control the virus replication. And then it took him a few years to then develop a knockout mice, which is the VCP KMT mice. And you could see that this is fairly recent data, which is should be completing this soon. He also found the same phenotype in the infected animals, where the knockout animals had significantly reduced joint swelling. So this is one example where, by controlling one of the host factor that's important to enhance virus replication, it could be a potential therapeutic as well. So I'll move on to another part of the question, which is also through the understanding of chikungunya pathology, which is led by another senior postdoc, CY, who has just recently won one of the early career research grants to look at this specifically, which is a vector-based approach by targeting mosquito saliva. So this was one of the earlier projects that was done in collaboration with DBS in NUS and also with Duke NUS, and where they are targeting the specific mosquito saliva proteins to control the pathogenesis. So what they have found was a particular mosquito saliva protein called cellokinin, which when CY then investigated this in the monocytes and macrophages, she found that the co-expression of cellokinin together with a virus actually would restrict the the monocytes activation during the virus infection. So she's continuing and also completing part of this study where she put this through into the animal model. And again, you could see that using the model that we have established earlier on, the cellokinin could actually reduce the not just the viremia, but also the joint swelling in the infected animal. So again, this is another potential new approach where look at host-directed therapy to control the virus-induced inflammation. So I'll end off with a last part, which in our quest of both understanding the mechanisms and pathogenesis, how could we then use this as a target to understand, to develop new therapeutics as well. So this was one of the very earlier work that my lab has done after we have established a mouse model. That was a former PhD student, Teck Hui, where he was trying to understand the role of T cells in virus infection and pathogenesis, and he showed that the CD4 T cells were the ones that were potentially controlling the joint swelling, because the knockout animals no longer have any joint swelling. And through understanding some of these, he also showed that when he was targeting the T cells with some of the clinically approved T cell immunosuppressive drugs, he found that when he tried the various, some of these drugs, such as rapamycin and fingolimod, of course, not everyone works, but one specifically, the fingolimod, which been clinically used as to control T cell migration. So it's categorized in the what we call the T cell migration blockers. He found that all the animals actually had a reduced joint swelling, and when he looked at the cellular infiltrates in this in all these cells, they also had a lot less pathogenic T cells. So this more or less, again, it shows how through understanding the pathogenesis, he uncovered the important cell types, and then by targeting these cell types, he could control the a disease outcome as well. So with this understanding of the importance of T cell, then it led us to another question where the distribution of malaria and chikungunya, but why did we want to even look at this? Now, if you look at this figure here, you could see from here that throughout the tropical belt between Southeast Asia, South America, and also Africa, you see that it also is a zone of the Aedes mosquito and also the Anopheles mosquito. And you can see it's interesting because in the next stage, actually there's a malaria talk going on at the same time. But this was the motivation for us to look at this in those days, where the clinical impact of co-infection was pretty much unknown. And there were some pockets of reports that actually showed the presence of antibodies against chikungunya in malaria-treated patients. So this was also one of the motivations that got us started. So in collaboration with Laurent Rainier, who actually is a known figure in mouse Plasmodium infections, we wanted to actually understand the impact of the disease pathology during the co-infection. So you could see that his group has established an experimental cerebral malaria model using Plasmodium berghei, ANCA, PBA. So in this model, all the infected animals actually suffer from mortality at the end, and also paralysis and all that. So, but what is more important was that using this model, they were able to show that the cause of ECM was actually highly dependent on the pathogenic CD8 T cells, and this was then associated with parasite sequestration in the brain. So by using this PBA model, then we actually attempted various methods of co-infection. And this is just one example using the concurrent co-infection of Plasmodium. We wanted to see the effect on the virus. And you could see from here that for animals that were co-infected with two pathogens, the joint swelling once again was very much reduced. Although the effect of viremia was there was no change. And just to shorten the story again, it took us a long while to understand the mechanisms behind what was it that actually prevented the reduced joint swelling. And what we found was that upon co-infection, actually suppress the CD4 T cell migration, and he actually uncovered it was due to the advanced cell death that the co-infection caused. So with that, we sort of again want to confirm the role of CD4 T cells migration to the joints, and also this will then prevent the joint swelling. So to take it off, you'll be asking, so what other questions are we looking at now? I think it's the various specific immune profiles. So if you remember when one of the earlier studies that we had done with the preclinical chikungunya vaccine, we found that the myeloid cell population was very important in controlling the disease outcome as well. So Fook Moon, which started this work also some years back, and he's about to complete this fairly soon now, where he then went on to study in depth the specific profiles of the infected joints. So using the CyTOF myeloid panel that was established some years back at the Singapore Immunology Network, he actually found that when he compared between non-infected and infected animals, there was one particular cluster that was quite unique. And obviously this was also the cluster that carries the pro-inflammatory monocytes. And specifically, one of these was he found a high expression of the CD64 markers. So you could see that it's very, very pronounced. And so then the quest to really understand actually what happens, which happens first, is it this or that then drives the CD4 T cells. So right now, he's actually trying to complete this part of it, where it is to understand the specific immune interplay between the CD64 positive macrophages and also the CD4 plus T cells. And so with the various depletion of the CD4 T cells in the wild type mice, you could also see that the phenotype is very different. But what is more interesting is that when he put this through, he isolated the specific subsets and put it through RNA sequencing. Specifically, you could see from here that between the depleted and non-depleted portions, the profile is very different, and specifically he showed that it affected the activation of the T cell profile and differentiation. So we are still trying to wrap this up, and the interesting questions would be, through this interplay, is it pro or is it is there any specific way that we could actually control the disease outcome specifically? And I'd just like to stress that for studies like this, in fact, sometimes the timing is also very important. And this is where, how could we devise the best treatment strategy if we want to also control the swelling. So I would just like to end off my talk here, where there's all this interesting work that will be led by the various young leaders, which are really proud of them, to continue with their own respective research questions. But also I want to acknowledge all the collaborators, both locally and also worldwide, who have been very kind and generous with the sharing of reagents, viruses, and also knowledge that enabled all this various work. So the question continues, could we, how are we able to make use of some of this relevant mouse model to understand vector-borne diseases in humans? So with that, I will thank you for your attention.
Thank you, Prof. Lisa.
Okay.
Yeah. We have a few questions. So one of them is, how representative are mouse models of human vector virus interactions? Because Dr. Julian Tang asks, the thing is that human immune responses are developed over one's lifetime, so definitely more complex than the genetically modified mice. So, how do we actually interpret, or how do we extrapolate it to?
Yeah, I think that's a very, very relevant question, I think, especially for clinicians, because they will see that obviously the mouse is not the human. Now, I just want to stress that indeed the immune system is actually very different for the mouse and the humans. But in terms of the general immune subset populations are quite representative, I have to say. It's not 1 equals to 1, not, but it is close enough to understand the mechanisms and some of the pathways. I just want to stress that the mouse models that I shared, that I presented today, none of them are immune compromised. All these are wild type animals, so it's as close as you can get.
Okay.
If you were to use the immune compromise, then obviously then there could be, it will help you answer that specific molecule, that specific pathway, right? But not the whole disease on its own. But so to make use of the mouse model, it's to understand, to reconfirm some of the importance of this immune cell or particular host factor that could have a very big role in controlling the virus replication and disease phenotype.
Right.
So it will also provide a preclinical means to also do some of this preclinical assessment of some of these possible therapeutics.
Right. So basically, as a starting point, I mean, you need a starting point for everything.
Yes.
So you would understand that, and then maybe build on it and go subsequently into the humans.
Yes, you definitely need an in vivo host before you go into a larger. Obviously, the non-human primates would be another way, but then that is also it's very high-end kind of experiments, and you don't use that model to confirm a molecule. Use that before, yeah.
Right. So do you foresee a vaccine for the chikungunya?
Oh yes, I think there's a lot of interest. I think that if you were to look out at the publication, there are many preclinical chik vaccines, and actually some are already on trials and some look promising. So I'm very sure that there's a lot of interest, specifically with the success of the mRNA vaccines over the SARS-CoV-2. So people are relooking, using that platform to look at all the previous relevant diseases that have been around for some time.
Yeah, so one single vaccine for dengue, tick, all of them would be great.
I'm sure Ing Yong will give you more insights into the dengue vaccines that he has been working on. Yeah.
One question, I know we didn't touch upon the Zika, but I'm pretty sure a lot of people will have a lot of questions about that. So how prevalent is Zika here in Singapore? To be honest, clinically, and now, a few months ago or a couple of years ago, maybe everybody was talking about Zika and we were trying to send off specimens, but now I hardly do. I don't at all. So, yeah.
I think the question is whether are we looking for it, right? Because if you say clinically, it's actually quite similar. I think everyone, the moment you start to have fever, I think the first test everyone is looking now is not dengue or not. I think it's COVID. I think, yeah. But I mean, the question is, I honestly think that it is around. It's just that it's so mild. I think it didn't cause any specific alarm, and but if you really go down and screen for it, when you try to hunt for it, I'm sure that you will see it.
So that brings us to the other question. The Zika association with microcephaly.
Yes.
I think that's its claim to fame, so to speak, right? I mean, when during the Brazil outbreak, everybody was so nervous about that.
Yes.
And subsequently, I think the studies were all very iffy. Okay, in other parts, Fiji had an outbreak, and then they said, okay, maybe not microcephaly, etc. Yeah. So, in your lab, or do you see that the Zika has a predilection for neural tissue, or it's just because of the huge numbers that we saw in Brazil?
Okay, that is a very interesting question, also very important. So admittedly, a few years back when Zika was the in disease, then I think there were many groups that launched into this, especially the microcephaly. Now there were reports that showed the virus could actually infect the brain cells and things like that. So I think, but the method of virus inoculation in these mouse models also matter. So in my lab, we did not directly look at microcephaly, but we have shown and we have published this was that in the infected mice, potentially, I mean the infected pregnant mice, there is a higher proportion of this mothers, I mean the mice, that will actually have fetuses that are smaller than the non-infected. But you need to generate a very big number. It's not 100% all the time.
Of course.
But then there is a higher proportion of this that cause. So what is the cause? We did not complete that, but I know there were various groups that have showed various possible pathways that how it could actually break through the blood-brain barrier. But I think none is really that conclusive. I would say that then there will be some other papers that will come in to actually counter that. So I think it's still a bit blurry.
Yeah.
But I guess, if we were to specifically identify the target cells which some group have shown, and also the exact pathway and how it actually can go up, I think that will be the interesting question. I think that one, it's not been answered.
Because even though chikungunya is so much common when we see these huge outbreaks, and even with dengue, I don't think we have seen any of these.
No. Although there have been some clinical associations from some other countries that also try to point to a neuro outcome of it, but still a little inconclusive. And we also have to be very clear, are all these clean, specific arboviral infections? Is it only that? Or could these patients also have some other ongoing conditions? That also we have to be very careful.
Great.
Yeah.
Thank you so much. And it's been a fantastic talk.
No, thank you. Thank you for having me.
Yeah.